Hello, everyone, and welcome to phyloseminar.org. Our current theme is the adequacy of phylogenetic models. In this series of 3 talks, we have been hearing 3 perspectives on model adequacy. First, we heard from Jeremy Brown about the role of model fit in resolving the tree of life. Last time, we heard from Sebastian Duchesne on model adequacy in infectious disease phylodynamics, and today we hear from Sarah Hilton describing her work on the model adequacy of experimentally informed site-specific substitution models. If you want to ask a question, you can either tweet at phyloseminar or type your question in the live chat box to the right of the video on YouTube. As I mentioned, today's speaker is Sarah Hilton. Sarah is a graduate student in genome sciences at the University of Washington, Seattle, doing her PhD with Jesse Bloom at the Fred Hutchinson Cancer Research Center. Welcome, Sarah, and thank you so much for participating.
Thanks, Andy. So today I'm going to talk about work from the Bloom Lab developing site-specific codon substitution models, which are informed from empirical measurements from the lab. I'm also going to talk about how we evaluate these models and ask whether or not the experimental measurements are kind of doing a good job of describing natural sequence evolution. So since I'm going to spend the majority of the time talking about substitution models and how we evaluate them, I thought it would be good by starting off by taking a step back and discussing models more generally. Biology is full of incredibly complex processes. You have metabolism, the immune response to infectious disease, social dynamics, and of course, evolution. And while we're able to observe the output or the data, we often don't know exactly how the data generating process works itself. However, we can use models, simple statistical expressions, as a tool to illuminate and understand the process. Now, approximating a complex and complicated process is not easy. And due to the necessity of simplifying assumptions, it will always be that, an approximation. So the challenge is balancing the simplicity of the model with its usefulness in describing the important aspects of the process. That is, reflecting on the assumptions of your model and identifying assumptions which may be impeding your understanding. Some models and associated assumptions that we spend time thinking about in evolutionary biology and phylogenetics specifically are phylogenetic substitution models, which aim to describe the forward evolutionary process of genetic sequences. So here, as an example, I'm showing the phylogenetic tree of seasonal H1N1 influenza virus from the introduction into the human population in 1918. So this year marks the 100th anniversary of the 1918 flu pandemic. Its evolution throughout the human population until it was overtaken by another strain in the 2000s. So in order to understand how the flu proteins evolved over this time period, we can take sequence samples and then build models to describe the changes between the 2 sequences. We can describe these changes and the underlying process on the nucleotide, codon, or amino acid level. So here's an example of such a model, the GY94 model from Goldman and Yang, a very common codon substitution model. You can see that this model describes evolution as a function of whether or not the change is non-synonymous or synonymous, a transition or a transversion, as well as the nucleotide bias of the gene. And while these parameters are certainly capturing some of the dynamics of the evolutionary process, there are also aspects which aren't accounted for due to these simplifying assumptions. And the GY94 model actually shares assumptions with many other substitution models. Specifically, 2 of the most common assumptions in phylogenetic substitution models are that sites evolve independently and that sites evolve identically. And there's a great deal of evidence that neither of these assumptions is true. However, I'm going to spend time talking about the second assumption, the assumption that sites evolve identically, and I want to do that for 2 main reasons. The first reason, as I'll show in the next few slides, is that the violation of this assumption of identical evolution across sites is immediately apparent when you look at protein alignments. And the second is that we can actually make strides to relax this assumption within the framework that we use for current models, which assume uniform evolution. And neither one of these points are true for the independence assumption. So when you have this kind of clear problem and a possible solution, it's a good place to start. So here are one of these multiple sequence alignments, kind of showing that the sites do not evolve identically. So this is just a snippet of a multiple sequence alignment from influenza virus hemagglutinin, where each row is a sequence sample and each column is a site in the protein. And now we can certainly see patterns in this alignment. For instance, some sites do look identical. Here are 2 sites that have only isoleucine. Some sites are perhaps constrained to the same degree. So here I've highlighted 2 sites which appear to be mutationally tolerant. They've sampled many different amino acids. And some sites, even though they don't look identical, seem to prefer similar amino acids. So here are 2 sites where you see a lot of glycine. But taking a step back and kind of looking this overall, it's clear that the sites in this protein are not evolving identically. This multiple sequence alignment is certainly not an even distribution of amino acids. So in order to capture this important dynamic of protein evolution, we need to build models which relax the assumption that sites evolve independently. That is, we want a model which accounts for these site-specific amino acid selections and preferences. If in our sequence we have a one nucleotide change from one codon to another, we want the model to be able to differentiate between a non-synonymous or a synonymous change. But if the change is non-synonymous, we want to be able to differentiate between whether or not the new amino acid is a tolerated amino acid or a deleterious amino acid. And what I mean by this is you could imagine that there's some change, perhaps isoleucine to glycine at site 65, where with this new change, the protein is still able to fold and perform its function. But you could also imagine that the same change somehow breaks the protein. It's no longer able to fold or function. And so we would call this a deleterious amino acid. Now, the first layer is relatively easier because the genetic code itself is not site-specific. Whether or not a nucleotide changes the amino acid encoded by a codon is going to be the same no matter what site you are in the protein. And most common substitution models, like the GY94 model I showed before, account for this layer using a parameter called omega, which is the rate of non-synonymous to synonymous change. But whether or not an amino acid is tolerated or deleterious is going to be site-specific. This means that the number of parameters in the model is going to balloon because we now have to account for each amino acid at each site in the protein. So that's 19 parameters for each L site in the protein, assuming that the preferences should sum to 1 at a given site. And so the question becomes, how are we going to determine these amino acid changes and the selection on these changes? And so one method might be to infer these preferences or fit them to the natural sequence data. However, this immediately poses the problem because if you have a multiple sequence alignment, it's simply a sample. If you don't observe an amino acid at a given site, is it because it's not tolerated or because it was not sampled? For instance, a tolerated amino acid could fail to be sampled because either the protein hasn't kind of explored that part of evolutionary space, or it has and we simply didn't sequence it. There are models trying to infer these amino acid preferences from these natural sequence of alignments, taking this into account. I've highlighted 2 at the bottom, but this is certainly not a trivial problem. An alternative strategy is to directly test the effect of a mutation in the lab. Rather than waiting for evolution to sample an amino acid, we can just make the variant in the lab and test the fitness effect. We can determine whether or not an amino acid is tolerated or deleterious empirically. Up until recently, this approach has had a scale problem. The experiment that I cartooned here, and kind of all of the experiments that you would have to do to measure these different amino acids, was very low throughput and would take a very long time. But in recent years, there have been technical advances which allow us to scale up this assay and kind of test all the effects of these variants. This is a new assay called mutational scanning. So mutational scanning is a high-throughput functional assay which allows us to measure the effect of every single amino acid change in a protein. Briefly, you can take a wild-type gene and introduce all of the single amino acid changes. You then grow up a library of protein variants. So in the Bloom lab, we study viral evolution. So most likely, in our experiments, this is a library of viruses where each virus carries a different variant. You then impose some sort of functional selection, and then using deep sequencing, you can count the initial variants and the selected variants and infer what we call this map of amino acid preferences. So I'm showing this map as a logo plot where each column is a site in the protein, and the height of each letter is proportional to the preference for that amino acid at that site in the protein. And this is a broad overview of the experiment, but I want to zoom in on a couple stages in hopes of just building a little bit more intuition. So the first part of the experiment is making all of these genetic changes. We take the wild-type gene, and then using PCR mutagenesis, we can make every single amino acid change away from wild-type. So at this first protein position, we would make every change away from wild-type methionine for a total of 19 variants, where the first site is different and the rest of the sequence is wild-type. And we can do this for every site in the protein, so that the total number of variants would be 19 times L sites of the protein plus 1 wild-type. So for a more concrete example, if you have a 500 residue protein, this is 19 times 500 plus 1, or 9,501 variants that you're making. And just a point to make here, demutational scan is now only going to measure the effect of these amino acid changes in a single genetic background, that of the wild type that you choose for the experiment. So next we take this library and we grow up these protein variants. So here we're showing these viruses. So you'll have variants which are the wild type sequence. You'll have them where it's 1 amino acid away at the first site of the protein for all the different sites in the protein. And then these variants are pooled together in a single tube, and this pool is selected for functional variants. So in the Bloom lab, these libraries of viruses are most often selected by growth in cell culture. That is, to be considered a functional variant, the virus has to be able to enter the cell, replicate its genome, and egress from the cell. But this is an important part of the experimental design. The results of your experiment are going to be dependent on the selective pressure used, and this experiment should attempt to capture the most important selective pressure you think is acting on this protein in nature. For results, you might expect the wild type to do quite well. You might expect to see some variants which do better than the wild type, some which don't grow at all, to be these deleterious amino acids, and kind of a whole other range of outcomes. And while thus far I framed the assay as an assay to study viral proteins, deep mutational scanning is a flexible assay which can be used for many different proteins utilizing different selective pressures. So here I've highlighted just a subset of the published deep mutational scanning sets, and you can see they're in different proteins using different selective pressures and even in different cell types. So once again, after functional selection, we use deep sequencing to count the variants and infer the preference for each amino acid at each site in the protein. And if we zoom in a little bit more on this preference set, we can see that the deep mutational scanning data looks similar to the patterns of evolution we saw in that multiple sequence alignment. There are constrained sites, there are mutationally tolerant sites, but importantly, the effect of every amino acid has been measured. So in that matrix of 20 amino acids by L sites in the protein, we don't have any missing data. And just to underscore the size of this experiment, here's the full data set for the influenza virus protein PB2, a protein which has just over 750 sites. And so you can see that there's a lot of information about the site-specific preferences of the protein here. And so if we return to our schematic or simplification of a site-specific model, previously the worry had been how to define these site-specific preferences. However, deep mutational scanning gives us a direct measurement of these parameters, and so this issue begins to fall away. That is to say, we can model the selection on amino acid changes without fitting to or inferring from the natural sequence data. And so that's exactly what we've done in the Bloom lab. We've incorporated deep mutational scanning measurements into codon models to describe site-specific amino acid selection. This allows us to relax the assumption that sites evolve identically, which we think should build better and more useful models of protein evolution. And we call these models Experimentally Informed Codon Models, or ExpCMs. And so today I'm going to talk about how we formulate these models, and then I also want to talk about how we evaluate these models. We've used traditional model comparison techniques where we compare the ExpCM to a model without site-specific selection. And we've also started to develop model adequacy tests as well. These evaluations are obviously important, as you should always evaluate your models, but I think it's particularly interesting in our case because we have these models where the minority of the parameters are fit to the data and the majority of the parameters come from these lab experiments. And so these ExpCMs gives us a tool to bridge the gap between what we know about selection on a protein in nature and what we know about selection on the protein in the lab. So our experimentally informed codon models follow the mutation selection formulation first proposed by Halper and Bruno in 1998, where the rate of change from codon X to codon Y at site R is described as a function of two processes, mutation and selection. And I'm not going to spend a lot of time talking about the mutation term other than to comment that it follows an HKY85-like term, but I am going to spend a decent amount of time talking about this selection term because that's where the amino acid preferences come into play. So I think it's useful to first just think intuitively about how we would use deep mutational scanning preferences, the data we get out of these big experiments, to describe selection on change. So once again, here's a snippet of the results from the experiment. And so at a constrained site like this, we would expect selection against any change away from the highly preferred glutamic acid. And so we want these changes to have a low probability in our model. We expect them to be purged. On the other hand, at a mutationally tolerant site, we wouldn't expect to see selection against any change because all amino acids seem to be roughly equally preferred. And so we'd expect all these changes to be roughly equally probable. And just to introduce some notation, we refer to the value represented by the height of each letter as the preference for that amino acid at each site and denoted by pi. So pi RAX is the preference at site R for the amino acid encoded by codon X. So here's the full formulation of the selection term F. You can see that when it's a synonymous change, it takes on a value of 1. When it's a non-synonymous change, the value takes into account both the preference for the starting amino acid, pi r a of x, and the ending amino acid, pi r a of y. There are also two other parameters in this term, omega and beta. Omega, like many codon models, is the rate of non-synonymous to synonymous change. However, in this model, it's the rate after taking into account the site-specific amino acid preferences. That is, you expect some rate of change given the mutational tolerance of a site, and omega either increases or decreases this rate. Beta, or the stringency parameter, rescales the preferences to reflect the difference in the strength of selection between lab and nature. And I'll go into beta in a little bit more detail in the next few slides. Omega and beta are not site-specific, and they are some of the few parameters in our model which are actually fit to the data. So as I said before, beta, or the stringency parameter, rescales the preferences. And so when beta is equal to 1, the preferences in our model are exactly equal to the empirical measurements from the lab. However, when beta is fit to be greater than 1, you see this kind of exaggeration of effects where the large amino acids get larger and the small amino acids get smaller. And so we would interpret this when beta is fit to be greater than 1 to say that selection in nature favors the same amino acids as selection in lab, just with a higher stringency. And this is because beta does not change the rank order of the amino acids. On the other extreme, when beta is fit to be 0, the preferences become flat. We've now lost all site-specific information and amino acid-specific information. And so we would interpret this, when beta is fit to 0, to be that selection in nature prefers different amino acids than selection in the lab. And in essence, our ExpCM model has collapsed down into a GY94 model. We can visualize the relationship between the preferences, beta, and the selection term. So here on the x-axis is simply the ratio of the preference for the ending amino acid, A of y, to the preference for the starting amino acid, A of x. So on the right-hand side, we have changes to more preferred amino acids, and so these are changes we would expect to be selected for. And then on the y-axis, we have the selection term, with higher values indicating a stronger selection for the change. So when beta is equal to 1, we see this positive relationship that we would expect. There's no rescaling, and our preferences are exactly equal to the empirical measurements. And then as we change beta, we can just see that this kind of selection strength is modulated. So when beta is greater than 1, selection for favored changes and against disfavored changes is stronger. But when beta is less than 1, selection for or against any change is weaker. And so this reflects the relative flatness of the preferences and our connection to the stringency of selection. So the resulting stationary state of the ExpCM is site-specific. So here are visualizations of the stationary state of the GY94 model and the ExpCM model. In the GY94 model, each site is exactly the same, and the differences from the amino acids are just due to the structure of the genetic code and nucleotide biases. However, in the ExpCM, there's the site-specific stationary state defined in part by these deep mutational scanning preferences, and so you're not assuming that every site in the protein is evolving identically. So after we build these models, so this is the final ExpCM and how we incorporate our deep mutational scanning measurements to model selection on amino acid changes. And so next I'm going to talk about how we evaluate this model. I'll show you some results from model comparison techniques we've been using and some of the questions we can answer with those. And then I'm going to talk about future work that we want to do using model adequacy tests to evaluate these models a little bit more. So as an example, I'm going to walk through a set of analyses for the influenza virus protein hemagglutinin, or HA. So HA is one of the surface proteins and is responsible for the virus entering the cell. So in this test case, we're using influenza sequences which have been circulating in the human population. This means that the selective pressure on the virus are all these complex parts of the infection process, some of which I've listed here on the left, but certainly there are more as well. And what we're saying with the ExpCM is that we can model all of this selective pressure using just cells in a petri dish. And so thinking back to this balance between simplicity and accuracy, we think we're moving towards a more accurate model because we're modeling site-specific constraint. But we need to make sure that our way of modeling this constraint, these cells in a petri dish, are not in of themselves too simplistic. The argument for this working is that the ExpCM, that the deep mutational scanning is measuring some of the major selective pressures on the protein, its ability to fold and function. And so you can see that a little bit by thinking about the viral life cycle. Here's a crystal structure of HA. The head is shown in red, and this is where the receptor binding site and the active site of the protein is. And then in this diagram, you can see the huge conformational change HA undergoes during entry into the cell. And so you can see it kind of springs up. There's this lever-like function to bring the membranes together, and all of this has to happen in order for the virus to complete its life cycle. And so I just wanted to point this out because if we're going to measure the selective pressure and the constraint on this protein, we want to model all of this. We don't want to just take one conformation, one protein structure, and kind of measure deviations away from this. We want to take into account everything that the protein has to do to be successful. And that's exactly what demutational scanning does. So as I mentioned before, in order to be a functional variant, the virus has to be able to enter the cell, replicate its genome, and egress. And so while we're not kind of getting demutational scanning preferences at each of these conformations, our results at the end are a little bit of a holistic look at what amino acids allow the protein to go through all of these different changes. And so this would be an argument for why, even though cells in a petri dish are very different than human beings, we might be at least beginning to model some of the main constraints on this protein. But on the flip side, as I said before, the vast majority of these parameters are these lab measurements and not from the data. So if you have an ExpCM with 500 sites, only 6 parameters in our model will be fit to the data, and 9,500 will come from the lab. And so we want to make sure that a model with so many empirical measurements really does do a good job of describing what's happening in nature. So to talk about the data itself in a little bit more detail, there are 18 influenza subtypes, HA subtypes. I'm showing the tree here. And other than that influenza-like HA, all 18 subtypes have a very similar structure and perform a similar function. So if you get flu this year, you're probably either going to get H1N1 from this side of the tree or H3N2 from this side of the tree. And even though both H1 and H3 can infect humans and cause disease, they're only about 42% identical on the amino acid level. So these two homologs are incredibly diverged from each other. And in the Bloom lab, we performed demutational scanning in both the H1 background and the H3 background. So the H1 work was done by a former graduate student, Mike Dowd, and the H3 work was done by my current lab mate, Joo-Hae Lee. And as I mentioned before, demutational scanning is only measuring the effect of these amino acid changes in one genetic background. And so because these two proteins are so diverged, we might not expect these scans to be exactly the same. So we can build these ExpCMs using the two different data sets and look at how well they describe natural sequence evolution. So the first set of analyses I want to talk about is testing the importance of our site-specific constraints. So we want to know how well the ExpCM defined by the H1 preferences, whether or not this is a better model than the site-uniform GY94 model for H1 evolution. And we can ask the same question for the H3 preferences. Is the ExpCM defined by the H3 preferences a better model than the GY94 model for H3 evolution? And so here are the results for the H1 analysis. We're going to look at 4 different models. We have the ExpCM defined by the H1 preferences plus or minus a gamma distributed omega value. And we have the GY94 model plus or minus the gamma distributed omega value as well. So this gamma distributed omega is a way to account for site-to-site rate variation, but it itself is not site-specific, nor does it affect the stationary state of the model, unlike the preferences. So we can see that both ExpCMs are better descriptors of H1 evolution than the GY94 models, at least as evaluated by delta AIC. We can also look at the fit parameter values, specifically omega and beta. So the GY94 model doesn't have this stringency parameter beta, but for both of the ExpCMs, the beta value is fit to a value greater than 1. So we'd interpret this as selection in nature prefers the same amino acids as selection in lab, just with a higher stringency. Or as a visual, here's a snippet on the left of the preferences unscaled, so beta equal to 1, and here they are rescaled with a beta equal to 2.01. So we can also look at these fit omega values. Omega values are used to look for signs of diversifying or positive selection, which is especially useful for bioproteins. And so this is usually considered when omega is greater than 1. For the models with the gamma distributed omega, I'm showing the mean values of 4 discretized bins of this distribution. And previously, it has been observed many times that omega values tend to be quite low for the GY94 model, representing the fact that these proteins are under purifying selection, and we see that here in our results. However, the ExpCM omega values are all higher than the GY94 models. And in fact, we have one omega value here, which is significantly greater than 1. And this is because the omega value for the ExpCM is the DNDS rate after accounting for site-specific constraint. And so it appears that modeling the site-specific constraint gives us more power to detect positive selection in these sequences.
Sure. I was also, have you considered also accounting for rate variation among sites with gamma distribution, and could that play a role in these preferences?
Sorry, can you say that again?
Variation in rate of substitution across sites. Is there any suggestion that that also might make a difference here?
So other than the gamma distributed, you're saying?
Yeah, so not just gamma distributed rate heterogeneity for this omega parameter, but or are you accommodating variation in substitution rates across sites here too?
Yeah, so I think the omega value, right, is the substitution rate across sites, kind of the number of non-synonymous to synonymous changes, so at least on the amino acid level. And so that is this kind of raw rate parameter is being distributed across sites. And then what we're saying is that if you're at a kind of mutationally tolerant site, you might already expect a decent amount of change, but at a conserved site, you might not expect a lot of change. And so the omega value can be a little bit higher because you're already like accounting for the fact that that site probably won't change that much just due to the kind of inherent constraints. Does that make sense?
Thank you.
Yeah, so overall with this analysis, we see that the ExpCMs are better descriptors of natural sequence evolution than the GY94 model, at least as evaluated by delta AIC and these other criteria. So it looks like modeling site-specific constraint is important, which is unsurprising, but also that these deep mutational scanning measurements are able to capture some of the constraint. And we can look at this a little bit more by examining two control models for the ExpCM, an averaged and a randomized control. So the randomized control takes the deep mutational scanning preferences and swaps them across sites. And so technically, we still have a site-specific model after this procedure, but we no longer believe that's an accurate site-specific model. We no longer believe that there's any reason for selection in nature to prefer these amino acids. And then the second control is to average the preferences across sites. That is, the preference for leucine at each site is the average of all leucine values in the experiment. And so here's what the values look like for the H1 data. And I've shown a line at 0.05, which would be what we would expect all the amino acids to be if they were all equally preferred. That's definitely not what we see. We see that proline has on average this very low preference value, and leucine on average has a quite high preference value. And so what this control model tells us is like we're still extracting some real information from the experiment. We just no longer have site-specific terms. And so if we want to see how well our deep mutational scanning measurements do, how well they are at modeling the site-specific constraint, we can add these two models to our table. So here they are here. Both the ExpCM average and the ExpCM randomized perform worse than the GY94 model with this gamma distributed omega. And specifically, you can see that both of the beta values are fit to be less than 1. So we'd interpret this as selection in nature, preferring different amino acids than selection in lab. And in fact, if you look at this randomized model, the beta parameter is fit to be basically 0. And so you would get out flat preferences from this model, and it has basically collapsed down to being the same as this GY94 model. And so what these control models tell us is that the improvement comes from site-specific constraint measured in the lab and not just kind of random constraint noise. You really do need to measure or at least take into account the constraint on each site. Okay, I won't go into as much detail, but I just want to show that we get similar results with our other deep mutational scan in the H3 background. So to hit some of the highlights, as we saw before, both of the ExpCMs are better descriptors of natural sequence evolution as evaluated by delta AIC. The stringency parameter for both of the ExpCMs is greater than 1, indicating that nature prefers the same amino acids as selection in lab, just with a higher stringency. And then finally, the omega values for the ExpCM are higher than the omega values for the GY94, as these represent the DNDS rate after accounting for this site-specific constraint. And so what we found is that the ExpCM defined by these deep mutational scanning preferences are better descriptors than the GY94 model in these matched analyses, where we're looking at preferences measured in the same background as our tree. However, these two deep mutational scans and these two diverged backgrounds gives us an opportunity to examine how well conserved these site-specific constraints are across evolutionary time. So on one hand, H1 and H3 have a similar form and function. You might expect that the site-specific constraints and selection remain relatively constant over time. On the other hand, these genetic backgrounds are only about 42% identical on the amino acid level. And so any effects contingent on the genetic background, these epistatic effects, would not be the same between these two scans. And so we think of these epistatic effects as shifting amino acid preferences, where the preference sets don't stay constant across the tree. So as a cartoon example, you can imagine a protein with a glycine at the first position and an alanine at the second, and maybe an A to V mutation at the second position breaks the protein. However, you could also imagine that the same mutation, A to V at position 2, is tolerated if the identity of the amino acid at position 1 changes. That is, the effect of an alanine to valine mutation at position 2 is dependent on the amino acid identity at position 1. And demutational scanning would only measure this effect here, where the first identity stays the same, and you would never recover the result in the second scenario, at least not with a single SAN. So obviously what this is really getting at is this assumption that sites evolve independently or that sites don't evolve independently. And our model does not explicitly relax this assumption, but by comparing ExpCMs with preferences measured in different genetic backgrounds, we can begin to get a feeling of the effect of these shifting preferences. So we can add another question. Are both the ExpCMs better than the site uniform model for H1 evolution, or is it just the ExpCM informed by the H1 preferences? And so here's the same H1 tree, but with two new models. Here are the ExpCMs with the H3 preferences. You can see there's a little bit of a splitting of the difference. So the ExpCM with the H3 preferences are better than the GY94 models, and their beta values are greater than 1. However, both of these models are worse than the ExpCM performed by the H1 preferences. So it does appear that form and function of the HAs is significantly conserved, such that a deep mutational scan in a diverged background can still do a better job of describing natural sequence evolution than the site uniform model. But we also have evidence of these shifting preferences and epistatic effects. And so I think this is interesting and kind of opens up a whole new slew of interesting evolutionary questions. How far across a tree can we stretch a preference set? At which sites are these differences most important? However, these evolutionary questions that I just outlined, as well as perhaps evaluating the models overall, are perhaps not best done by model comparison methods. So in order to truly investigate some of these questions, we have to move towards a model adequacy framework. And while I've been thinking a lot about this, this next section is not going to have as much results as more as just an extended future directions and discussion. And so the model comparison techniques are not quite the right tool for answering some of our questions, because model comparison is more like a game of would you rather. So asking, is the ExpCM better than the GY94 model, is like asking, would you rather fight 1 horse-sized duck or 100 duck-sized horses? We could spend hours debating the relative merits and different metrics and et cetera, but at the end of the day, neither model comparison nor would you rather allows you to answer neither. That is, we don't want to just ask if one model is better than another model without acknowledging the fact that they could both be bad models. So we want to shift our analysis towards asking questions like, is the ExpCM an adequate model of H1 evolution? And more specific questions like, at which sites is the ExpCM an adequate model of this evolution? And for our site-specific models, this site-specific evaluation is important. There might be sites in the protein where the site-specific constraints and selection from all these factors is adequately modeled by growth and cell culture. So you can imagine this might be true for sites which are very important for protein folding, for stability, for receptor binding, et cetera. Then there might be other sites where it's just too simplistic of a model or too artificial of a model, and these might be where we start to pick up some inadequate sites. So I'm lucky to be the third talk in the model adequacy series, so these tests have been well introduced. Here, I'll just briefly go over how we plan to use the model adequacy test for the ExpCMs. So we're working in a maximum likelihood framework, and therefore we're closely following the parametric bootstrapping method from the Goldman-Cox test from Goldman 1993, but we are using a slightly different test statistic. So first we fit an ExpCM given some natural sequence alignment and our deep mutational scanning preferences. We then simulate sequences under the ExpCM. So then we have to take a test statistic from the natural alignment and the simulated alignment, and that test statistic should have some connection to the part of the model we want to test for adequacy. And since what we really care about is the deep mutational scanning measurements and their ability to describe the site-specific constraint on a protein. So we've decided to use a distance between the stationary state of the model and the amino acid frequencies and the alignments. And so then we can compare the distance from the natural sequence alignment to the distribution of distances from the simulated sequences. And if the distance falls within this distribution, we would consider it adequate. And if it falls on the extreme, we would consider it inadequate. And importantly, we can do this calculation and get a p-value for each site in the protein. And so this allows us to do our site-specific analysis. And of course, by saying distance, I am just kicking the can down the road. We have to come up with our actual distance measures themselves. And I'm putting distance in quotes because one of the metrics I'm going to talk about is not a true distance. But we've decided to start off by calculating the Jensen-Shannon distance and half the sum of absolute differences. So you can imagine that there's some alignment frequencies from either the simulated alignment or the natural alignment. And then this would be a representation of the stationary state of our ExpCM at some site. And so here, because the stacks are quite similar, you would have a small distance. And then as the stacks become more and more distinct from each other, our distance would get larger and larger. And in case anyone is curious, here are the formulas for those two metrics. And there are certainly other distance-like metrics, and we may expand or contract our list in the future. But the important part is that the part of the model that we're trying to interrogate, the part of the model that we kind of care about, is that site-specific stationary state defined by the deep mutational scanning preferences. And so we want to come up with distances and statistics that allow us to test that part of the model. And so purely hypothetical data, we would hope to come up with something where for each site in the protein, each metric that we choose, we have some p-value and we can call whether or not we think the site is adequate or inadequate. And so what can we learn about proteins and protein evolution from these adequate sites? So on a purely phylogenetic side, we want to make phylogenetic inference from adequate models. But I think what's interesting about these ExpCMs is that we might be able to learn about selection on these specific proteins kind of more broadly. So if we care about selective pressures on some sort of protein, as I've talked about, we can do this by looking at what's happened in nature, by looking at these natural sequences. And we can do this by looking at the lab and performing all of these measurements. And these two methods are independent of each other, which is very powerful. So we can learn a lot from each of these two sources. But what this ExpCM does, it allows us to link these two sources together. And where they disagree, where the ExpCM is inadequate, might point to interesting differences between selection in lab and selection in nature. And if we think about these inadequate sites and we think about what we know about what we measured in lab and how our experiment was designed, we might be able to rationalize these differences and learn some more things about the protein. And so when I've been thinking about potential results and kind of where these differences or inadequate sites might be, I've kind of been bitting them into two main categories. I imagine you could pick up sites where selection in nature is different from selection in the lab. And you could also pick up sites where there's some selective pressure in nature that you're simply missing in lab. And so to kind of explain how, in my mind, these are different, here's an example from the H1 data from the Bloom lab. And what Mike and Jesse found is by just looking at their preferences from the lab and the frequencies in nature, they found this one site where nature has a very high preference for serine, and the lab had this high preference for tyrosine. And what they found was that this site was important part of the cleavage and activation in HA, and specifically the genetic background they used was this lab-adapted strain called WSN, and it had actually changed and evolved to use plasmin to activate and cleave HA instead of a protease like it happens naturally. And so this is an example where somewhere in the experimental setup, something got just a little bit too artificial. Now the selective pressure is different, and that's why you might have, hopefully an adequate site in future analysis, but at least by visual inspection, inspection, you can see this. And this would be in contrast to something like this study here, where when they were looking at an enzyme, they performed the selection using 3 different substrates. And so here, these scatter plots are simply the correlation between the preferences in these different scans. And so you could imagine you could use these model adequacy tests and these ExpCMs to ask which of these substrates is kind of the best selective, or represents the selective pressure in nature the best. And you could also imagine that there would kind of be different sites that were adequate or inadequate depending on which of these substrates you used. And all of this is important both for understanding the selective pressure and the kind of evolution of these proteins, but also people want to use these deep mutational scanning sets for engineering proteins, interpreting clinical variation. And so you could imagine that if we kind of quality control these preference sets by looking at how well they describe selection in nature, might help improve those analysis down the road. So overall, site-specific constraint is an important part of protein evolution. I hope that I've been able to show that we can actually model a lot of this site-specific constraint using empirical measurements from the lab. And that because these empirical measurements are independent of our natural sequence alignment, we can explore questions about how these preferences shift over time. And in the future, using model adequacy tests can help us determine which of our lab measurements are adequate describing this evolution, and that inadequate sites might point to some interesting biological mechanisms. So thank you all for listening. Thank you to my mentor, Jesse, for Juhei and Mike for making the data that I presented today, my funding, the Noun project for many of my images in my slide design, and Eric and Andy for inviting me to speak. Thanks.
Thanks, Ed. That was really informative. I also think it was really nice to see, from having had more previously hypothetical discussions about why we might want to interrogate specific aspects of a model, to this very concrete idea of not only do we want to interrogate these specific aspects, but we have very interesting hypotheses that can be generated and addressed by doing so. And in that vein, I was wondering if you were thinking about things like comparing adequate and inadequate model-specific sites for like H1 versus H3, so different assays in different parts of the tree, and seeing if they share similar adequate sites and if that might be interesting or meaningful.
Yeah, no, definitely. I think that's really, when we did this model comparison analysis with H1 and H3, I think that's kind of immediately the question you want to ask and the questions that people do ask us. And so I think the model adequacy tests and pinpointing exactly inadequate and adequate sites will help us get to that. I think it could say a lot of interesting things about kind of how the proteins fold, because while they're very similar, there are some slight differences. And so maybe we can identify some of those sites. And then I think the other important thing is that, there are ways to kind of just compare these deep mutational scanning sets and look for shifted sites. But I think the extra layer that we get by doing these model adequacy tests is not just saying that the sites have shifted, but that seems to matter. And so it's just that like extra layer of context.
Yeah. And sort of speaking of does it matter, I was wondering if you had any sense and could elaborate on other sorts of effects that you might expect from inadequate or less good models. So you talked very nicely and clearly about like what happens to this omega selection term when we have models that don't account for the site specificity. But do you know if these inadequacies propagate into other aspects of the models, like tree topology inference or branch length inference and other things that they might affect?
Yeah. So yes, my guess would be yes. I don't have any data or concrete examples to say that. One thing that I'll say is that currently all of our analysis is done with a fixed tree topology. And so while we kind of have this feeling that it probably is going to affect tree topology, and others have shown that site-specific constraint or modeling or not modeling it can affect tree topology, we don't really have a good way in our current framework to address that, though that would definitely be something we would want to do in the future. As far as branch lengths, we have shown previously, and others have shown that modeling this constraint definitely affects branch length, specifically your ability to estimate the length of longer branches. And so you can imagine that sites where you have inadequate models are going to affect branch length and might disproportionately affect these longer branches on the tree.
Cool, thank you. That was very informative.
So, I mean, I can imagine situations in which you had a site that sort of was well represented by the ExpCM for part of the tree, but maybe not for the whole tree.
Mm-hmm.
I mean, do you think it would ever be possible to sort of test some sort of subset or?
Yeah. So, one thing that we would kind of want to do for the H1 and the H3 is to test out the ExpCM on kind of progressively more diverged trees and see, are there sites that remain adequate or inadequate across the whole tree, or is there a point where like you've moved so far away from your focal sequence, like where the scan was done, that you've started to have these inadequacies. We don't really have a good plan as of now other than identifying them to like model that better necessarily. But I think kind of the first pass would just be kind of looking at different tree sizes and different divergences and see how our results change.
And can you also walk us through sort of the pipeline? So do you think that this would ever like feed back into your cell culture experience? I mean, I guess that you gave a pretty good example of like how it was sort of changing, but I mean, I don't know much lab biology. I mean, is it actually realistic that you would say like, hey, whoa, look at this site and then identify that, okay, instead of using protease, it's using this other thing, and then you could actually update your lab protocol based on that?
Yeah, so as also not a lab biologist, I don't know if I can speak very confidently to like the number of inadequate sites that we would be able to kind of nicely rationalize like Mike and Jesse did for that one site. But I think as far as that, there's like two ways you could go when you have that information from the WSN. One could be is like, all right, we won't use that strain, we'll use a different strain, and like it might take a little bit more work to get that up and running, but maybe we think it's worth it. And then I think another thing is just, we measured 564 sites. If you can say like this one site isn't great, we just won't kind of highly weight it in downstream analysis. I think that's another thing that you can get out of this. So rather than having to go all the way back to the drawing board, say, okay, it looks like 95% of our sites are, this is all hypothetical, look pretty good. This one site just seems to be something was different between lab and nature, and kind of use that information moving forward.
That's interesting. So just for instance, like can you just explain what are the downstream analyses that you would be able to accommodate that sort of thing?
Yeah, so I can imagine like there's one where like people want to use, or there's a lot of work on using kind of these demutational scanning sets for interpreting clinical variation. So if you have a patient coming in and you sequence that, or you sequence this gene and you see this variant, you want to be able to interpret the variant as far as like would it be deleterious and would it be tolerated. And so while we don't have a human HA gene, you could imagine like you wouldn't want to use the lab data on this kind of artificial change to interpret a clinical scenario. And so, or if you're designing a protein that is going to be out in nature, and you want to design it based on these demutational scanning preferences, if we've identified that one of these sites is possibly inadequate, maybe you don't want to kind of weigh the demutational scanning too much in your kind of redesign of this protein. So very hand wavy and hypothetical, but that's kind of what we're thinking.
Cool.
No, I think that's an interesting way forward. Well, with that, I think it's time that we all thank Sarah again for participating very much. That was a very informative presentation, and we will see you guys again for Phyloseminar in 2019.
Yeah, next one will be about weird phylogenetics, like not just looking at DNA sequence data, but like expression data, hopefully, and CRISPR arrays and all sorts of craziness. All right, thanks.
Thank you.